Maybe some of these will work.
You want me to go back there?
No, I'm going to put my notes. And we'll give out the test. Okay, good morning. If I could have your attention, please. So, just a couple of announcements. I'm going to give your exams back at the break. The grades were, as you noticed, on Piazza were significantly higher. That's really good. That rarely occurs on the second exam, but it did, so I was very pleased about it. And the people that are in here apparently are in here to stay. So, going forward. For the graduate students, you do not do that exercise on the initial contribution. That's just for people doing posters. And remember, that deadline's been moved, not this Friday, but the following Friday. And we'll also, next week, we will also have a training session about how to do a PowerPoint poster. And so I'll ask a couple of members of your group to go over to the Teaching, Learning, and Professional Development Center and go through that training so that you can bring that knowledge back to your group. So we'll do that probably it'll be next Thursday. It's usually Thursday at 4 p.m. So all of your group can't go because they can't fit into that room, but at least 2 or 3 can go. Okay.
Next Thursday at what time?
4. Yeah, and I'll go over that. I'll post it on Piazza and everything. It's in the library, and then you may know where it is already. Okay. Seemed like there was something else. I'll think of it probably. Okay, so where we had gotten to in moving proteins through the secretory pathway is we've got them in the ER now. What do you do to them? Well, one of the big things that you do is you glycosylate them. And there's a couple of different types of mechanisms. So we've got to be able to distinguish between 2 big types, O-glycosylation and N-glycosylation. Okay? And then ultimately, we're going to ask the question, how does the ER understand when to get rid of a misfolded protein? Because one of the big functions, as you know, of the ER is quality control. And so you try to find misfolded proteins at that point, and you don't waste any more energy moving them through the secretory system, and you eject them out of the ER. Okay. So let's first deal with why would you want to put sugar groups on a protein? Well, typically, where do you put them? You put them on the exoplasmic domain of that protein. If it's a secreted protein, you can put it all over the protein because the whole protein is going to be ejected and go out. A lot of hormones that come from your pituitary, like FSH and LH, those are all heavily glycosylated, and that keeps them intact and stable. It prevents proteases from chewing on them. So that sugar coating, you can think of it as a way, we sugar coat things to get it through your stomach. Different drugs have different types of coating. Well, that's the same reason you sugar coat proteins, is to protect them from degradation. And actually that sugar group is actually part of its 3-dimensional structure, so sometimes it's actually recognized by the receptors to which those hormones bind. So protein stability is an important reason. Actually, we do put sugars on groups just to ask the question, are you folded properly? And we'll look at an example of how looking at sugar groups tells you whether it's folded or not folded. Protein targeting, we'll look at a very specific example in lysosomal enzymes. Obviously, there's a big penalty for sending a lysosomal enzyme that chews up stuff to the wrong compartment. So we'll look at how you target each lysosomal enzyme to the correct place, which is the lysosome. And then we won't go into this very much, but those sugar groups on different membrane proteins actually link to other proteins that have sugar groups. And so there are protein-protein interactions that are facilitated by sugar groups. So cell-to-cell adhesion is an important example. Okay, so how do we distinguish different types of glycosylation? We'll talk about the simplest is O-glycosylation. And why do we call it O-glycosylation? I don't know if you can see this very well, but let's, a couple of examples. If the sugars, and that's represented by these little hexagonal sugar groups, are attached to an amino acid through an oxygen atom, it's called O-link. It's very straightforward, okay? If it's attached to a residue on asparagine, then it attaches to a nitrogen atom, so then, therefore, it's called N-linked, right? Okay, so is that the only difference, the type of amino acid to which you can attach these. That's not the only difference, okay? There's some generalizations. They don't always, those characteristics are not always explicitly true, but we can make some generalizations. Notice that the amino acids available for this are not only the straightforward ones that you would think of, serine and threonine, if you're going to make an O-linked amino acid or sugar group, but lysine. Now you'd think, well, that's an N-link, right? No, all N-link have to be linked to asparagine. There's no such thing as linking to any other nitrogen-containing amino acid. But what happens is you can take lysine and you can post-translationally hydroxylate it. So lysine can become hydroxylysine because an enzyme, after the protein is translated, gets hydroxylated. And that provides an oxygen atom to which O-linked sugars can be attached. Okay, so there's serine, threonine, hydroxylysine, hydroxyproline. So proline has the same sort of enzyme that can attach a hydroxyl group after the protein has been made, right? Okay, one of the things you notice about O-linked sugars is there's not very many of them. Rarely are there very many long chains, okay? And the reason for that is a single enzyme has to attach every one of these sugars one at a time. So it takes a lot of time. The other thing you'll notice is they tend to be linear sugars. They're rarely branched. This is probably the most branched O-link that you'll ever see. But this is really unusual to have any branching. So it should be small and linear. So this is a very uncharacteristic O-link sugar. Usually they're very short and unbranched, okay? And they're attached to an oxygen atom. So now we're coming up with some generalities that describe them. Also, the other generality is the major sugar groups are attached one by one. Each different sugar gets its own enzyme. So the enzyme that attaches a mannose to this is a very different enzyme than one that attaches galactose or N-acetylglucosamine. Okay, and we're not going to memorize all these sugars. We do need to know a couple of their names because they have certain roles, particularly glucoses and mannoses. Okay, the other difference, the other group is N-glycosylation. So you're going to attach it to the nitrogen atom of the asparagine R group, right? And it has to have this consensus sequence. So it has a consensus sequence is a sequence that regularly compels the protein glycosylation machinery to put a sugar group here, all right? And so this is always the case where it's an asparagine followed by any amino acid except proline, all right? Prolines are always kind of weird. And then the next amino acid is either serine or threonine, except you're not putting the sugar group on them. You're putting it on this asparagine. And look at this thing. This thing is highly branched. What else do you notice about it? There's lots of residues, okay? And there's lots of, we call this high mannose number. There's lots of mannoses. I think there are 9 mannoses in that little cluster. So this is really an oligosaccharide tree. And that's what's attached in block that is all together in one step is stuck onto that amino acid. The other cool thing about this is that this is attached co-translationally. What that means is the protein is being extruded through the translocator. As soon as it emerges on the luminal side, and as soon as this consensus sequence appears, it immediately gets stuck because this sugar group is going to help it fold properly, and it's also going to protect it. Okay, so a large distinction between N-glycosylation and O-glycosylation. All right, so we're going to look at detail some of the steps that go into N-glycosylation. This is probably the most important slide in terms, it sort of summarizes things because obviously you have to make this sugar tree somewhere, and I'll show you that. We're going to look at it, but you're not going to memorize how you do that. But then there's a lot of construction that goes into making this tree, and it actually starts out in the cytoplasm. But let's pretend that we're further along than this. We've already constructed the tree. We've got to link it to something. There has to be a carrier, and that carrier is called dolichol. This has got rubber tails. It's got the isoprene units.
that make it sort of rubbery, and it spans both leaflets of the ER membrane. And it exposes on the luminal side a couple of pyrophosphate, 2 phosphates, and then linked to that is the oligosaccharide tree. And then what happens is, if this is an exposed consensus sequence with the proper asparagine, then that is recognized by an enzyme called oligosaccharyl transferase. And that tells you exactly what it's going to do. It's going to break off that sugar tree and stick it on that amino acid. And that happens while this thing is being extruded into the ER. So as that loop is coming down, this thing is this enzyme is attaching the sugar to it, and you've now N-glycosylated that protein. But one of the things about N-glycosylation is that the cell is never finished. It's like an irritated sculptor that's always sculpting something and removing the nose and putting a different nose or different eye back on it. It always is picking at this thing. So it'll cut off. It starts out with 3 terminal glucoses, and one by one those get cut the longer this protein stays in the ER. That's sort of a timing mechanism that it's going to use to see if this thing is folded properly. The other thing it's going to start doing is it's going to chew away on some of these mannoses. So the second way that the ER knows how long the protein has been in the lumen is how many mannoses are left in the sugar tree. All right? So there are timing mechanisms. And if it overstays its welcome, it gets ejected. All right? All right, so again, I mentioned that I would just briefly talk about the biochemistry. You know, there's no reason to learn all these steps. You know, you can go look it up if you're excited about it. But dolichol is constructed this way, usually in the cytoplasmic phase, where these enzymes one by one put a phosphate, and then you add sugars and mannoses. And when you get to a certain size, you've got 5 mannoses and some N-acetylglucosamine. It actually is flipped over. So there's a transport protein. It's not flipase, but it's related probably to the ABC transporters that can take this big thing and move it across the lipid bilayer. So you would have to have a protein do that. And so it flips it, and now that sugar tree is exposed on the exoplasmic side where it's supposed to be, and so you can create the final dolichol molecule that's fully loaded with the oligosaccharide tree, and so then it can be transferred to the protein. But it starts in the cytoplasm, it ends in the ER, all right, ER lumen. So we attach this thing to the protein, and what are we going to do with it? Okay, so what happens is as you come off of that, it's released by that enzyme, the protein tries to fold, right? And some, about 55% of the proteins fold right the first time. They just get it right. Or they fold a little bit, but they stay on the folding pathway that you looked. And others are not quite so nimble. They're a little bit clumsy. And what happens is, if you cut these glucoses, the longer you stay in there, and if you have just one glucose, then you are a target for this protein here, which is called calnexin. And calnexin is a membrane protein. And what it does, it's called a lectin because it binds to sugar residues. Proteins that bind to sugar residues in the terminology of biochemistry are called lectins. So this is a lectin binding protein, and it binds to the protein, it recognizes, or it binds to that protein, it's a little unfolded, so what it does is this is a chaperone protein, and it's going to recognize that terminal glucose, says, I have to hold on to you because you're not fully folded. What do you think it's actually going to be looking for? If it's unfolded, or if it's not folded properly, what will be exposed that shouldn't be? Hydrophobic groups, right? So it probably has a hydrophobic binding site, and so it grabs there, it recognizes, there's one sugar group, you're misfolded, because the way to get out of here is to get all the glucose is cut off. So it gives it a chance to refold, and then this glucosidase cuts that off, and that releases it from calnexin. And calnexin, it then has a chance to ask itself, am I folded? And if it's fully folded, it exits from the ER in a vesicle and goes on its merry way, which we're going to start covering today, okay? So the next step in the journey is generally to go to the Golgi, and not just anywhere in the Golgi, but the cis Golgi. Okay. Okay, so what happens if this is still misfolded? This protein recognizes the fact that it has no terminal glucose, but it's misfolded. So again, it has a way of recognizing exposed hydrophobic regions, and so it binds to it, and then it retags it. It goes, you're not ready to go. And putting that tag allows it to go back through this process and try to refold. And so after a certain number of times, what's going to happen is if this comes out misfolded and it has a certain number of mannoses that have been pulled off of it, it gets ejected. It says, we tried, but you have failed. That happens to about 15% of the protein. So what happens to it? Okay? It's removed back through the translocator by a mechanism we're not going to worry about, but it uses the same protein translator. It can go this direction as well as out. And it goes through unfolded, okay? And then what happens is two things happen to it. Number one, you don't want to destroy this nice sugar group. So you remove that sugar group by this N-glycanase. So it just chops off oligosaccharide, and you can retrieve those sugar residues and reuse them, right? So that's not wasteful. The second thing that's sitting here is something called a ubiquitin ligase, okay? And so when this misfolded protein comes through here, you remove the sugar group and you attach a ubiquitin. Where do you attach them to? You attach them to lysines, if you want to know. And it's a very specific place. It's called a destruction box. And so, but here's the key. You don't put one ubiquitin, you polyubiquitinolate it. So you put a ubiquitin and then you attach a ubiquitin to a ubiquitin, so you get this little chain of ubiquitin. So that's a signal to the cell that this is time to destroy that protein. So polyubiquitin sequences attached to a protein means you send it to the proteasome, which is a really cool machine that will take this protein, and one of the reasons it's cool is it's constructed, we know its structure quite a bit, and it has this chamber into which the protein is pushed. And what that chamber does is it removes the ubiquitin. So it knows better than to chop up the ubiquitin. So that gets ejected when it gets into this little cap area. And then the rest of the protein is threaded through this, you can think of it as like a paper shredder. And it shreds that protein into individual amino acids, and you get to reuse those. Okay, so you don't really lose a lot of biosynthetic stuff. Okay, so ubiquitin can go on for another round of tagging misfolded proteins. So what's the point? This is quality control. You do not want to have misfolded proteins accumulate into the ER lumen. All right? Are there any questions so far? Things that don't seem. Okay. So.
You do have a hand.
I'm sorry? Oh.
No, that mechanism was just for the
Yes. That's correct. But it's likely that most of the proteins are going to have an in-link somewhere on it. Okay. But this isn't the exclusive mechanism by which quality controls happens. It's the best understood. Okay. There is a buddy for calnexin. It's called calreticulin, and you don't need to really know it, but they're both called cal something because they're calcium sensitive. So the calnexin is that membrane-bound version of it, but there's a version that's free in the lumen. It's going hunting its own proteins. It's called calreticulin. So they're both chaperone proteins, and they're both lectins. Okay, now, so what happens is sometimes there's so many misfolded proteins that they tend to overwhelm the folding machinery inside the ER. Now, this is particularly true if you're infected with a virus, and that virus is making lots of its own little viral coat proteins, and it just loads the ER full of that stuff. And so the ER must have a way of detecting when it's being overwhelmed. And so it's going to have to ask for helpers. So there's several different mechanisms. They all work slightly different, but we're going to focus on this one that's called IR1. Okay. And as you can tell, this is a transmembrane protein. Typically, what happens is it's not dimerized. It's found in the monomeric state. okay? So what's going to happen is, here it is, this is IR1, and typically when it's in the monomeric state, it's bound to BIP, okay? If BIP is bound to IR1, it's not functional. It's turned off. This IR1 protein is turned off, okay? Now, what happens is, if you load this up with a bunch of misfolded proteins, you're going to need a lot of BIP, aren't you? So BIP will disassociate from IR1, and it'll try to do its job by binding to misfolded proteins. Well, what happens is that signals to IR1 that BIP is being really busy, and by golly, we need more BIP, okay? So what happens is, if it's empty here, this binding site for BIP is empty, it will dimerize with a buddy, okay? Another one of these IR1 that's doesn't have any BIP, and so they dimerize. And this protein has a couple of different activities. Well, it has three. First of all, it has BIP binding activity. That's the signal for, are you busy, are you not? Okay, if you're busy, it means you have BIP trying to help out and you need more BIP, okay? And the second activity is it dimerizes and then it cross-phosphorylates. It auto-phosphorylates, okay? But this one will phosphorylate that, and that will phosphorylate this. So it phosphorylates itself. And that turns the enzymatic activity on fully. And what is that enzymatic activity? It is a endonuclease. A riboendonuclease. It's looking for pieces of messenger RNA that it can cut, okay? And the protein it's looking for is very specific. It's called HAC1. HAC1, and that's kind of good because it's going to get hacked, right? What's weird about this is in the cytosol, and it has an intron that wasn't cut out. So apparently it snuck through the nuclear pore with that intron being assumed to be an exon, right? So the protein export machinery just says, oh, that must be an exon, so it sends it out, but really there's an intron that keeps this messenger RNA from being translated until it's needed. So how does it know when it's needed? Well, that has to be turned on, okay? And so IR1 is turned on, and so it chops that little intron out. So it chops it out, but you need another protein to ligate it, and your book is not happy enough to show you that, so I'll just tell you what it is. It's RIG1 phosphate, so it dimerizes. It gets phosphorylated by this thing. We're not going to worry about the details, but you need a ligase that sticks the two pieces together. So that happens here. It's ligated. So that was step 4. And now you've got a full functional messenger RNA. And that messenger RNA can be translated. You're in the cytosol, so you're already there. You pick up a ribosome, you translate it, and you produce the protein called HAC1. HAC1 is a transcription factor. Okay? And so you know how this thing's going to work, right? It's going to bind to, and so it's got to have a nuclear localization sequence. It's going to bind to an import receptor. It's going to crawl through the nuclear pore. It's going to meet up with RAN-GTP. That will expel HAC into the nucleoplasm. It will bind to its response element, and it turns on a series of 300 different genes. All of them are designed to help out with the ER folding problem. So it turns on lipid synthesis, so that's going to make the ER bigger, okay? It turns on protein synthesis of specific proteins like BIP and more IR1 and a lot of other proteins. So the best thing to think about is one of its major products is more BIP. And if you have more BIP, that's going to occupy these guys, and it'll turn off the IR1 mechanism. So it should be, it's a negative feedback. Produce more BIP, take care of the folding process, and get rid of the problem. Also, one of the genes it turns on is the cluster of genes required to make a translocator. So the translocation machinery is beefed up, you've got a bigger ER, and you got more BIP. So it's a really nice little system for detecting overload and responding to that problem. Right? But it's kind of curious how it happens. Right? Are there any questions about what went on there? Because it's a little multi-step problem. Okay. Okay, so I think that brings us to the end of that lecture. And so we need to go to 14. And that's good. So now we're into chapter 13 because this is a really complicated process what's coming out next. We've just done the very, very, very first step. We said, okay, you're made, we've stuck you into the ER somehow, you're folded properly, you're in the membrane or you're not in the membrane, but you're finished. We finished kind of fooling with you. That's actually not true because protein glycosylation starts in the ER, but it's generally finished in the Golgi. So what we have to do, everything else uses the same trafficking pattern, that is intracellular vesicular trafficking. So this is moving proteins by encapsulating them in a vesicle and moving them in a small packet from one organelle to the next. And it's really cool, but it's very complicated because this place is really packed together. And somehow you're going to have to do this, which is you have a donor membrane and you're going to have to capture the right cargo in that little vesicle. You're going to have to deform the membrane. So there are actually membrane deforming proteins that bend the membrane, okay, into a circle. That's called budding. So you have to coat it, and then the coating process actually produces the bud and generally includes proteins that snip it off. So you have budding, and then that buds off. The first thing that happens is you must then uncoat the protein. The problem is, if you don't uncoat the protein, you cannot see the targeting information that is displayed on the surface of the vesicle. Okay, so it makes a lot of sense. The coating process is to pull it off of the donor membrane and turn it into a vesicle. Then the coating falls apart and displays the targeting information, which makes a lot of sense. So in this case, it's not a nuclear localization sequence or a nuclear export sequence. It's a compartment targeting sequence that tells it, where do I go next? And typically the next stop is to the Golgi. And not just any, you need to kind of refresh your memory about what the Golgi looks like. The Golgi has several different compartments. The one that's closest to the ER is called the cis-Golgi network. Okay? And so somehow it's going to fuse with that, and it's guided to that particular compartment, which is called the target compartment, by a series of proteins. Okay, so this looks simple, but it really isn't very simple. Okay, so that's where we're headed. One of the things that we're going to use to help us find the correct budding membrane and also to find the correct target compartment is the state of the phosphoinositides. Okay, what that is it's phosphatidyl inositol, and that let's see if I have an example of that. Well, I'm not going to show you that right away. It's a membrane lipid that we're going to get into, but that membrane lipid is varied depending on how many phosphates you add to it. Okay? And these are always found in the cytoplasmic leaflet. The head group is a 6-carbon ring that we'll get to here in a moment, and it's got different places where you can attach phosphates. And it's like putting a digital code into the membrane lipid, like you need to go here. All right? So the state of this phosphoinositides helps direct the traffic that the membrane budded vesicle is going to go to. Right? Okay, so we're going to have lipid help to tell us where to go. The proteins, the cargoes that we're carrying inside of the vesicle, we have targeting information. Okay, so there's lots of targeting information to tell us where to go. Correct. All right, so this is the outline. So let's go back to, I thought this was 13. We don't want to do that particular coding mechanism first because it's very complicated. So let's go do a simple one first. All right, so let's talk about how you achieve specificity and the major types of coding, because the first step is what? Is to create a vesicle. And the way you do that is you butt it off by using the coating process. And there are 3 different types of coatings that we'll talk about. The best understood by far is clathrin. The second type is called COP1 and then COP2. The word COP comes from the fact that these are the protein components of this coating is a codomer protein. So that's where you get the word COP. Okay. And you use these different coatings to go to different destinations. So the state of the phosphatidylinositol is one clue, but also which type of coating is another clue about where you're supposed to send that vesicle. Okay. So let's look at the overall pathway. Here are our choices, and the way your book diagrams this is sort of like, again, a sort of British subway map. So we're here, there's really only one choice. You can go to the Golgi, which is the cis-Golgi network, which is this bubbly looking membrane right here. So this is the Golgi apparatus, and it's got a bubbly layer, and it's got these other sacs, which we call cisterna, okay, or cisternae. And so you have the cis-Golgi network, you have the cis-Golgi, the medial Golgi, the trans-Golgi, and the trans-Golgi network. So on both sides of the Golgi are these bubbly looking things, and that's because vesicles are fusing here and vesicles are leaving at that end. So the vesicles actually move through this system, all right, or the protein cargo. So here we are at the ER, and we're butting off all these things, and that's what's happening right here. So we can go to the Golgi, and the Golgi can send traffic backwards, and we'll talk about how does it know which way to go. If you're going forward, let's talk about some vocabulary. If you're going forward towards the plasma membrane, that's called anterograde. Anterograde vesicular traffic. If you're going towards the nucleus is usually right here because the ER is part of the nuclear envelope. It's called retrograde. So if you're going plasma membrane anywhere in this direction, retrograde. Anterograde is forward. Okay. So typically what happens is you go from the ER to the cis-Golgi, you go through all the Golgi, we call them the Golgi stacks because they're stacks of sacs, okay, Golgi stacks. You go to the Golgi membrane, and that's the trans-Golgi network, and that's where all the traffic, this is the big directional, this is like this huge railroad, Grand Central Station, that sends things to the correct destination. One of those destinations is the early endosome, right here. And then from the early endosome, you can go to the late endosome, and then from the late endosome, you always go one place, and that's the lysosome. Okay? But you can shortcut things. You can go from the trans-Golgi to the late endosome. So here are the compartments: ER, Golgi, early endosome, late endosome, lysosome, and then secretory vesicle. So they have to somehow have a way of telling which types of cargoes go in which place, all right? Okay, so here's what the 3 coatings look like. We can actually identify them in transmission electron microscopy. This one kind of looks like sort of coated like a soccer ball. It's kind of got lattices on there. The COP1 basically looks fuzzy. COP2 has a slightly different coating look to it because all 3 of these are using different kinds of proteins to produce the budded vesicle. Okay? We're going to focus on this guy because you probably even briefly studied it in introductory biology. Okay? But here's the basic rules with a few exceptions. If you're going from the ER to the cis-Golgi network, what you use to get there is COP2, which is diagrammed in red. Okay? So if you're going from here to there, that's what? That's anterograde. You use COP2. Once you get into the the Golgi stacks, whatever direction you're moving between the stacks, you use COP1. And that includes going from the cis-Golgi network backwards, retrograde, back to the ER. That's also a COP1. So there's really, the simplest one of these coatings that we only use in one situation is COP2. We only use it for going anterograde to the Golgi. All right, so that's real easy. Sort of the generalization, COP1 is sort of within the stacks, okay? But sometimes we use it to do what's called constitutive secretion. And what I mean by that is If a protein is made, and a good example of that is collagen. We just sort of, fibroblasts make collagen all the time, and they just secrete it without being told to do so. And so if you have that type of protein, it has no targeting information. It has no targeting information. And if you do not have any targeting information, what happens is you get produced. How do you get into the ER if you're going to be secreted outside? We know how to do that now, right? What's your signal, topogenic sequence? You only need one thing. Yeah, it has a very specific name. Signal sequence or signal peptide. Okay, so collagen must have one thing, and that's a signal peptide at the N-terminus. And so after that's chopped off, it has no other topogenic sequences, it has no other targeting information, and if you lack targeting information, you get a free ride to the plasma membrane. Okay? And so that's when you would use COP1 to get here, you'd use, COP2 to get to the Golgi. You go through these stacks by a mechanism that we'll talk about. But when you get to the trans-Golgi network, which is the Grand Central Station, the decision is made because you lack any kind of directional information to send you in a vesicle that's coated with COP1 to the plasma membrane. Go straight there, do not pass go, do not collect tube and collars. Okay. All right, so let's talk a little bit about clathrin because that's going to be our first example. And so clathrin is going to be used in very specific instances. One of the classic ones is actually retrograde traffic from the plasma membrane to the early endosome. Okay? That's the classic clathrin-coated vesicle. Okay? So going from plasma membrane to early endosome is a clathrin, and it's shown in green here. Okay? If you want to go from the trans-Golgi network to the lysosome, you have to go through the early endosome and late endosome first, and so that traffic is guided by clathrin. Okay? So if you're going to butt off from the Golgi, the trans-Golgi network, and your destination is ultimately the lysosome, you must use clathrin-coated budding mechanism. Okay? And then we'll talk about this rather complicated system here a little bit later. This is the regulated secretory pathway. We'll talk about that. So use this in very, very discrete situations. Okay. All right. Before we go into those details, what I'm going to do is we're going to take a break and give us a couple of minutes. So Bailey is going to put your exams by group, and so just hang back a minute, give her a chance to get those distributed in order, and send one person down to get everybody's tests. Okay. So it'll take a minute or two.
Professor, I have to leave in a little.
Yeah, thanks. But and if you come in late, always come in.
I know, I'm sorry.
Because I always turn around and look.
I'm so used to like, sitting there.
I know. Okay. Oh, but get with your group because we are going to do it. Theoretically. Well, you can use websites. No, so there really should be no
situation, you can't get over
So just go to the library and there.
Well, you don't have to go now, you just electronically get it. Did you go to his talk? What you do is you use, you have to, as a Texas student, you're allowed to register for it's called Iliad. So if you go to the library's website and find the Iliad thing, I mean, if you can't figure this out, just email the librarian. But you use interlibrary and get that question down, they will get back and get it on. Now, the other trick you can do is if you can find out, it doesn't always say that, but the corresponding author, you can email them. And 9 times out of 10, if you ask for a PDF, say, I'm interested in your work on blah, 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 be very nice about it. They want to spring. The more they send out their paper, the higher the impact. So you don't get it that way. I don't see that. I still have to sign it. But the website is the only site in the place, and that is the because that's the only allowable. Now, you can get familiar with the topics, like Wikipedia or any kind of website you want. It's just it's corrupt information. People put things in there that are wrong. And put things in there that they think are true, and it's sort of a work in progress, isn't it? But so that's why. I prefer to call it simple. It does. No, I find that once, so it has to have a certain value to be considered a secret. That's all it has. It's not going to stick in the brain. It's going to go into the yellow room. And if it has no other signal information, like it doesn't have a certain type of sugar, we'll talk about that. It's going to, if there's no other targeting information, it's going to be really sticky with all of these.
Bring you back pictures.
Yeah. Oh, yeah. Bring back the ones that are not picked up by your group. So anybody that's not here, you need to bring those back here, and I'm going to put them in a box outside my door. That's fine. It is a complicated, yeah. Let's see. Okay, why don't we get started again? Graduate students, you know that we have just one paper, it's the beta cell dedifferentiation paper, instead of two. So. That's Wednesday, 2:30, right? Okay, so Clathrin coating is our first mechanism that we'll look at in some detail. And if you look at these things, it does look like a sort of a soccer ball lattice. And the reason for that is that the proteins that make up that have this sort of structure. This is a heterohexamer. And so this is Clathrin, and it has two types of chains. It's got a heavy chain that makes up sort of the lattice work, so 1, 2, 3, you call this structure a triskelion, okay? And then it's got clathrin light chains that are actually calcium binding proteins, and they are attached to the heavy chains, okay? So we know exactly what this thing looks like. You put it together, and it forms these lattice-like structures. And so the purpose of this coating is to actually bend the membrane. So the act of this polymerizing to itself is going to pinch off a ball. Okay? Not exactly. It's going to form a vesicle. You actually need another protein called dynamine to actually pinch it off. Okay? So here's again, the basics. We're going to use this as our little model because this is very well understood. So we have a receptor. Let's use something that we already know about, the transferrin receptor. Single pass membrane protein. We know what it does. What's the cargo that the transferrin receptor is going to carry into the cell? Transferrin. Okay, so it's going to bind to that receptor, and that receptor is sitting here. So somehow it's going to change its conformation. So the cytoplasmic end of that receptor is going to change its shape, right? So that's going to recruit proteins that say, build me a clathrin vesicle. Okay, so that's the idea. And so we're looking at this step right here. That green line is a clathrin vesicle that's forming, okay? And here's some of the details. Your book is not very specific about it. It's one of the rare places where it's just a little iffy. Okay, so you have the cargo receptor, which in our case, let's use the transferrin receptor. The cargo is transferrin that's picked up from the blood. What's attached to the transferrin? Iron, right? Okay, so what's going to happen is that's going to change the cytoplasmic shape of this protein, and that's going to help recruit a protein to the cell surface. That protein is actually there's a protein called ARF, okay, ARF, and ARF is an old friend of ours. It is a small GTPase. Okay, so it's going to work when it's bound to GTP in the on state, and then it's going to fall apart and leave the membrane when it is bound to GDP. So it's going to go through that GTPase cycle. All right. So that recruits ARF here, and the big thing about it doesn't even show ARF. You can't even see ARF. All right. But ARF is one of the first things that has to bind here. And the reason it has to is that, you see this little thing, it looks sort of like a devil space? It actually, when we're going to get up and look at it closely, it actually looks like Mickey Mouse. It's got two little ear domains that are actually called ear domains. Those are called assembly particles, okay? And the assembly particle is a good name because it helps all of these components assemble together. And it's not just any assembly particle, it's AP2, assembly particle 2. So the job of ARF is to bind to the cell membrane, recruit the assembly particle, and the assembly particle does all the rest. Okay? It's going to recruit the cargo receptor, and it's going to recruit the clathrin heavy chain to it. All right? And what happens is that's going to form, when the clathrin gets all pulled to the membrane, it immediately starts to self-assemble. All right? And so it forms a clathrin coat, and that pulls the vesicle, it buds the vesicle. Okay, but it needs a little help cutting this little stem here. That's actually very energetically disfavorable to take these two lipid membranes. What's that? This is the plasma membrane, and push out all this water in the neck and squeeze it off. That actually requires another GTPase called dynamine. You can think of that, energy, dynamine, it's dynamic. It's actually going to split GTP. That's what pinches off the vesicle. And then this little vesicle floats away. What must happen? If you've created the vesicle, what's the first thing that has to happen after you create the vesicle? Uncoat it. Why? Display the targeting infusion mechanisms for its destination membrane. Okay, so it's very logical. You have a cargo. It says, I'm bound. Okay, I got transferrin. I'm going to recruit ARF, and ARF is going to come to the membrane. And so let's go through. We're going to look at a very specific example of this. That just shows the details of dynamine curling around here. And it's really cool because you can actually, if you put a form of GTP in there that cannot be hydrolyzed, what happens is it forms the neck, So the neck is actually formed when dynamine is bound to GTP, but the actual pinching process requires that GTP be split. And so if you give it a form of GTP that cannot be hydrolyzed, it's called GTP gamma sulfate. Then what happens is you form a bunch of vesicles attached to the plasma membrane by a long neck, but it can never bud off. And this is what it looks like. You get a bunch of these. They're frozen in the dynamine on state, but they can't pinch off because they can't split GTP. It's kind of a cool process. Okay, let's go back to what we're going to do is let's look at this video first, because there's a pretty decent one from your book, at least to get an overview.
Eukaryotic cells take in extracellular molecules through a process called endocytosis, in which the plasma membrane invaginates and pinches off cargo-filled vesicles. This movie shows a series of electron micrographs that have been artificially morphed to show the process of endocytosis as it may occur. The process involves a variety of molecules, including the cargo molecules that the cell takes in, the receptors that capture the cargo molecules, and molecules called adaptins that mediate contact between the receptors and the clathrin molecules that act to shape the vesicle forming at the plasma membrane. In the electron microscope, individual clathrin molecules can be seen as three-legged structures called triskelions. Each triskelion contains 3 heavy chains and 3 light chains. When a clathrin coat forms on a membrane, the globular domains that make up the tips of the heavy chains bind to adaptins, which interact with cargo membrane proteins. The assembly of a clathrin coat can occur spontaneously as numerous individual triskelions come together, interact through their leg domains, and ultimately form a closed cage.
Goal. Okay. All right. So that's pretty cool. As I mentioned, we know quite a bit about this process, and in particular, what's kind of interesting is several years ago there was a student in this class sitting in the back of the room. Usually, I don't know why, named Mike Henney, and he got his Bachelor of Science from here, and he actually had the good fortune to do his PhD at Cambridge in the McMahon lab, which is very famous for clathrin coating mechanisms. And so he did his PhD. He actually discovered a new protein that causes tubular sections of the ER and the Golgi to bend. It's called an F-bar, okay? And so he discovered that, and now he's doing his postdoctoral work at Cornell, and so moving right along through the system. And so I actually have visited his lab electronically a couple of times, and his mentor, Dr. McMahon, is actually giving a talk, a plenary lecture at the cell meetings this year in San Francisco. So I'm looking forward to that. I will probably be modifying my notes after I get back. All right, so you get the simple version now, fortunately for you. And let's go back. We're on lecture 14 now. Okay. And for some reason that's not showing up here. That clathrin coating stuff should have been. Let's go back. Surely I didn't skip over it. Okay. Well, we'll talk through it then. one of the things that you noticed in that video is that the heavy chain bound to something called an adaptant. Now, what's an adaptant? An adaptant is what makes up assembly particles. And your book doesn't really show it very well, but I'm going to show it to you. Okay. One of the things before we leave that particular thing, notice here, I talk about ARF here, and so we need to put all these different components, ARF, assembly particles, cargo receptors, together. So to do that, I'm going to draw you a picture because your book doesn't cover it very well. Okay, so let's turn this thing off. Come on. There, good. Okay, so the first thing that happens, of course, is that the cargo binds to, say, the transferrin receptor. It undergoes a conformational change, and that helps recruit a bunch of proteins. One of those proteins is actually ARF, okay? Now, what's cool about ARF is that it's in two different states. When it's bound to GDP, what it is basically it's a protein, and it has retractable landing gear. So it's got lipid anchors that are pulled inside. It's actually got one embedded alpha helix, just like prostaglandin H2 synthase, that's going to stick into the cytosolic leaflet when it's deployed. And the other lipid anchor is just a standard lipid phosphatidyl fatty acid anchor, okay, like SARC has. So it's got two different anchors. But these things are all retracted when it's bound to GDP. But what happens is if this goes to the plasma membrane, okay, and it's going to encounter its GEF, okay? So this comes and encounters the GEF. What is that going to do? It's going to release GDP, and it's going to pick up GTP. So when it's in that state, it now looks like this. It changes its conformation, and it has one alpha helix like this that's going to insert in, if we kind of make this really big, so this is the cytoplasmic leaflet, like that. So just like prostaglandin H2 synthase, it's going to take that alpha helix and go in sideways and kneel into the plasma membrane, okay, the cytosolic side. And the other one is displayed as, it's just a typical phosphatidyl fatty acid anchor, like sarcase. I think it's a myristoyl fatty acid anchor. So it's got two ways of holding on to the membrane. But the reason it looks like that is it's got GTP bound to it. Now, so that means ARF is in the active state. What does it recruit? This thing is then, the ARF is then going to recruit the assembly particle number 2, AP2. And assembly particles are made of adaptants. That's where that word came from. So we have to construct the structure of an assembly particle for you to understand what's going on. And again, what it looks like is a little bit like Mickey Mouse. It's got an ear domain like this, shaped like that. And so this is the alpha adaptant. Okay, this component here. It's got a little sort of wavy little thing, so it has a little tassel on it, so that that ear domain can move around and interact with different proteins. And this ear domain is going to interact with a couple of different partners. Right? Number one, it's going to interact with ARF. Okay, so this is all out. This is the ear domain of AP2, alpha adaptant. It binds to ARF, okay, which you know is stuck in the membrane now. So that is what recruits AP2 to the membrane. Okay. The second thing is it's going to bind to a lipid. It's going to bind to phosphatidyl inositol. What does that mean? Phosphatidyl, it means glycerol backbone, 2 fatty acid tails, and then the head group is inositol. Okay? Phosphatidyl inositol. Okay? And that's just a 6-membered ring. Okay? That's going to have phosphate groups. Let's see, 1, 2, 3, 4,
4 and 5. It's going to have 4 and 5 phosphorylated. And we have a name for this form of phosphatidylinositol. It's called PIP2 because it's got 2 phosphates on the inositol ring. It's so famous that we give it its own little name. It's PIP2. That phosphatidylinositol binding site is in the cytosolic leaflet. Okay. So Mickey Mouse here is going to hold on to the plasma membrane via 2 things. Number 1, the alpha-adaptin is going to bind to ARF. Number 2, the alpha-adaptin is going to bind to the lipid, PIP2. It's got this huge negative charge that it sees and it binds to it. Okay. Now, we're not finished with Mickey because the other part of, there's a couple of other pieces of Mickey. Let's draw those in there. And if you have an alpha-adaptin, you're probably going to have a beta-adaptin. Okay? And you do. It looks like the other half of Mickey. Okay? And it's got a little wavy section, and it's got an ear domain. This is, let's give ourselves some room here. This is beta-2 adaptin. There. What is beta-2 for? It binds clathrin heavy chain. Okay? So we're starting to bring all the players together. So beta-2 adaptin binds clathrin. What part of clathrin? Heavy chain. So now you have the components that you need to construct a clathrin-coated vesicle. But what else do you need? What are we leaving behind? Why are we forming this in the first place? The what? What's our example? Transferrin receptor holding its cargo. So something's got to recognize the cargo. And you don't recognize the cargo because it's outside on the other side of the membrane. So it must be able to recognize the cytosolic domain of the cargo receptor. And that thing is called mu. It's mu adapted number 2, mu2. All right? mu2. So mu2 binds to what? The cargo receptor. So if we were to sort of draw this, you'd have the cargo receptor up here somewhere. See, it's a single pass membrane protein. Let's put it right here. Okay? And it's got a extracellular domain. What's this? This is transferrin. This is the domain that's recognized by mu2. So Mickey sort of lays on his back with his little mouth and is bound to the cargo receptor. One of his ears is recruiting clathrin heavy chain. His other ear is holding on for dear life because he's being chased by somebody. But, you know, by PIP2 and ARF. So everybody's coming together, and it's all because of Mickey doing his job. Okay. And then there's another subunit that we're not going to say a lot about, and that's called the sigma subunit. And the sigma subunit is, we'll just put it right here. And so that's the sigma 2 subunit, and that's for structural integrity. That keeps Mickey together. Okay. So this thing really makes perfect sense. You've got something that grabs to the cargo, something that's hanging onto the membrane, and then when the thing starts to and then something is grabbing the clathrin heavy chain. When that polymerizes, it's all going to involute into a vesicle. And then we're going to use dynamo to pinch it off. Okay, how are we doing so far? Okay, so we've got 2 GTPase enzymes. One is ARF, the other is dynamo. And let's see if I have. I don't think I have the right PowerPoint to show you. There we go. Not sure what happened to it. Okay, so the end result of that thing went to sleep, didn't it?
the end result of that is you butt off this vesicle, but there's a really key thing that happens next, is somehow you've got to have this thing to happen. And we could talk about all the specific proteins, but I'm going to make it simple for you. And one of the things that happens is, what's happened to the light chain? So there's a clathrin heavy chain, there's a clathrin light chain. So the clathrin light chain is involved it is a calcium binding protein. So when this thing buds off, it changes, presumably, it changes the affinity of, here, let's draw the triskelion. Like this, and then like that, and that, okay? And then the light chains are here, right? These long-legged things, those are the heavy chains. So calcium binds to the light chain after it's been deformed into this ball, okay? So it must increase its affinity for calcium. And when calcium binds to the light chain, it tells it, recruit heat shock protein 70, okay? So the heat shock protein is an ATPase that's going to bind to an uncoated clathrin structure. So it peels off all of the clathrin heavy chains from the vesicle, which uncoats it and reveals its targeting information. So it's bristling with all these other targeting proteins somewhere in the vesicle. Okay. Also located, if you're for completeness sake, what else must we have in that vesicle? There must be a ARF gap located in that vesicle that's going to tell ARF to dissociate. How do you get ARF to dissociate? You tell it to split GTP, so that's what the gap protein does. It's the GTP activating protein. It tells ARF to go from GTP, which means its landing gear is deployed. It retracts its landing gear when it splits GTP and goes to ARF GDP. And then it will release into the cytosol and look for another binding site, right? So that closes out, so that unpeels the thing, and now it's ready to go to the target compartment. And in this case, since it's coming from the plasma membrane, it's going one place. That's the early endosome. Early endosome. Okay. All right, so this is a lot of steps, but this is the simple version. Actually, there's over 100 different proteins that come in here. And so we actually talk about this, we talk about clathrin and the assembly particle as being hubs. And they are the hub of activity that recruit dozens and dozens of proteins. And we still haven't put all of that together. Okay. But we're getting there. Or at least McMahon's group is getting there. Questions so far? All right, so that's what happens with one type. That's the simple type of coding and uncoding of a protein and its cargo. Okay, and the details, of course, are here in your PowerPoint. Let's see. So we have about 10 minutes, and we have so far no questions, so we will go to the next one. Come on. Well, what did I do with it? Sorry. 14. Okay. So this is what I'm talking about when I talk about, turn this, oops. This is what's called the clathrin interactome. And we're trying to put all of these different proteins together. Everybody is touching somebody and everybody's recruited at some point. But I've just given you the major players, which are, you know, AB2, clathrin and dynamine, and then heat shock proteins, 70, okay? So it's more complicated than this. Fortunately for us, we're not going to worry about it. Okay. This is what I wanted to show you. So, and it was in there somewhere. Again, at least in this textbook, this is not your textbook, so I had to get it from a different one. And the other problem with this is it's not showing the plasma membrane bringing in cargoes. This is the Golgi apparatus sending cargoes away. So don't worry about it. It's the same process though, because you see, here's the players, here's ARF. And what's wrong with this thing? Is that what the leg would look like? No, one of the legs is only going to go straight down. That's going to be the myristate fatty acid anchor, and it's only going to be embedded into one of the leaflets, the cytosolic leaflet. The other is going to be a alpha helix that's turned sideways that kneels into the cytosol. So this is sort of accurate in terms of protein shape, but it's not accurate in terms of the lipid anchors that it uses. Okay, that's a minor point. The point is, this is being recruited by the ARF-GEF, and we know what that is, but I'm not going to burden you with what that protein's name is. But this brings ARF here, and ARF's job is to recruit Mickey Mouse, which is AP. Now, it's AP2 if you're going from the plasma membrane and bringing cargoes into the cell. But if you're going from the trans-Golgi network outwards, you use a different Mickey, which is AP1. Okay? So you see all the specificity is generated by all these different elements, the type of coding, the type of adaptants, which direction are you going, and also the type of lipid, the type of phosphatidylinositol. So there's information there that tells it to do everything. All right? So you recruit Mickey. Mickey is going to bind to ARF. Mickey is going to bind to the membrane. Mickey is going to bind to the cargo receptor, which is this protein. We'll talk about what this is, but this is just a receptor. It's a cargo receptor. And remember, it's always binding to the cytosolic domain of that cargo receptor, because it can't see what's on the other side here. Okay? And then this thing peels off. Okay, and then that's a more accurate picture of AP2, and we've talked about this. Okay. And this is the diagram that I was trying to get to earlier. This is the phosphatidylinositol lipid that's found almost exclusively in the cytosolic membrane because it's for targeting information for vesicles. It's also, we're going to use it again as a signal transduction method. So this is we go a little bit deeper with this particular phospholipid because we use it a lot. So these legs are only embedded in the cytosolic leaflet. The head group sticks out like any self-respecting hydrophilic molecule. So this is hydrophilic because it's got a phosphate bridge, right? And then you can load this up in 5 out of the 6 positions with phosphates. And each one is going to look a little different and recruit a different binding partner. So what's recruited by PIP2? And this is a form of PIP2, okay? That is, which protein is recruited? Mickey Mouse, okay? And what part of Mickey Mouse? The alpha adaptant, okay? So one of his ears binds to this guy right here. Okay. And to ARF. Okay, so you can see, in fact, this is what Mike Kinney and Cornell was working on, this whole system. What do all these different phosphatidylinositols tell the cell what to do? And so when we're having to decipher that code, just like we're having to decipher the epigenetic code, and it's complicated. And this just shows the fact, so what kind of a membrane protein would this be? It's direct peripheral, because it's binding directly to a membrane lipid and not to a membrane protein that's inserted into the membrane, right? So that is, Mickey would be a good candidate to be that type of protein, but Mickey also binds to the cargo receptor. So it's a hybrid type of peripheral protein because it's doing this and it's binding to a transmembrane protein, which is the cargo receptor. So that's very tricky, okay? So it's a interesting type of hybrid protein. Okay, and we do not know the code yet, but we know some parts to it. So we know that if you're an early endosome, when you leave the plasma membrane, one of the things you're going to do is you're going to be converted to this green state, which is you're going to only have a phosphate at the 3 position. And then in order to go to the late endosome and get gobbled up, you're going to have a phosphate added to the 5th position. And then that's going to allow it to fuse with the lysosome, in which a third this, phosphate in the 4th position is added. So is there any reason to memorize this? No. It's just to show you that there are complicated changes in the state of the membrane lipids that help direct the traffic. And how does that work? It's because each lipid recruits a different type of protein. Okay. That's the concept. All right. Any questions so far? Okay. Okay, now, so when we come back next time, we're not quite ready to leave, just almost, we're going to talk about this because we know this type of trafficking mechanism very well. And so we're actually not going to ever talk about the details of the COP1, but we are going to talk about the COP2 because just to show you the difference between clathrin and COP2 is very, it's a very good comparison, but there are lots of similarities too. So, all right, we'll do that next time. Okay.
It's about 20 minutes away.
I've been there.
Okay, so the lab
Actually,
started
I'm in
a little later, and so because of that, we might be late. Depending on when I get there, if you want me to come, I guess depending if I'm here by, if I can get here by 2:30,
Right. I mean, if you within,
like 10 minutes of that. I just don't know what time it's end because it depends on
That's the way
how
we're
scans
supposed
leak. Yeah, you know, that's just how they go.
yeah, if it doesn't work out, just let me know.
I will.
Shoot
I'll
me.
still send you questions.
Question.
But if I can't come, then that's why. But I'll email you the same day.
Okay.
So I forgot that I had to do that tomorrow.
Okay.
Oh, maybe it has one of those things.
I know. Tell me
I
about
forgot to
it.
drive is longer than what I'm used to. So.
It is.
Thank you.
Sure.
Does that have